Okay, what we're going to cover briefly is blood, and this is mostly for the lab section. So, what I'd like for you to know, generally speaking, at this stage in the game, because we still are early on in anatomy and physiology, just basic function of blood, right? Your heart is going to pump blood, and when we think of blood, primarily we're thinking about red blood cells. But we have to also remember there are white blood cells. Red blood cells are known as erythrocytes, and they're going to be carrying oxygen and gases and carbon dioxide and nitric oxide. Um, Blood will distribute nutrients to all the cells and tissues of the body. They're going to carry and transport hormones to different cells and tissues and organs of the body, and they will transport waste products. They can help get rid of toxins from the body. They're going to regulate pH um, of the body. They can act as a defense mechanism, not red blood cells, but blood in terms of white blood cells, in terms of our defenses. So when we think of our immune system, we're thinking of white blood cells known as leukocytes. And our bodies can also produce antibodies to antigens, right? The antigen is the foreign protein. The antibodies are going to attack that. So we are going to talk a little bit about RBCs, which are red blood cells, WBCs, which are white blood cells. Red blood cells are also known as erythrocytes, white blood cells, leuko's white, leukocytes. And then we'll also talk about platelets briefly, which are involved with clotting. So when here's our red blood cells, which are erythrocytes, white blood cells, which are leukocytes for the immune system. We'll talk about the 5 different types of white blood cells, and then platelets, which used to be called thrombocytes, not thrombo, that should be a T, thrombocytes, like a thrombosis, which is involved with clotting. So when you go to the doctor's office or you go to a lab, they typically will do a blood draw, right? And when they take blood, they're not going into an artery, they're going into a vein, right? That's called a venipuncture, puncture, vein. And they're going to spin it through a centrifuge. And when they spin it, all the heavy stuff drops. So all the cells and the formed elements drop to the bottom, and then the lighter substances go to the top. So on the formed elements, we're talking about red blood cells and white blood cells. On the top, we're talking about plasma. And this is why blood is considered connective tissue, because plasma is the extracellular matrix. These are the cells. So remember, epithelium is when you have lots of cells closely packed together, like the tiles on a floor. But when you have some cells scattered here and there with lots of extracellular matrix, and here the extracellular matrix is plasma, that makes up 55% of blood, whereas the smaller portion is the cells, the red blood cells and white blood cells. So when we look at blood and we break it down, we'll see the formed elements are red blood cells, white blood cells, and platelets. And the red blood cells make up are almost 100%, right? It's 99.9%. Your white blood cells less than 0.1%, and platelets less than 1.1%. But that makes up the formed elements. Your plasma, which is the matrix, the extracellular matrix, makes up 55% of blood. That's water, right? That's why you got to drink water. You take your body weight, cut that in half, and drink that much in ounces. That's how much water we should be drinking throughout the day. Then we have plasma proteins like albumin and fibrinogen. And then there's some other solutes that we have in blood. Okay, so that gives you a little bit of a breakdown where it's some cells with lots of matrix, which is why blood is connective tissue. It's just considered liquid connective tissue. Okay, now let's look at the blood cells themselves, okay? So the red blood cell doesn't have a nucleus. Other cells do. We'll see white blood cells have nucleus, but not the red blood cells. Because if you think back to the cell that we did before and all the organelles in a previous lecture, you can see that the nucleus is a large organelle. It occupies a lot of space within the cell. We don't want the red blood cell to have little space because the nucleus is occupying it. We want the red blood cell to carry hemoglobin. Heme, globin. The heme is the iron containing and non-iron containing portion. And then the globin is a protein. So many things are transported around by hooking up to a protein. Hemoglobin is one of those things. We want this erythrocyte, this red blood cell, to carry oxygen. And oxygen binds to iron. That's why when people are iron deficient or they have iron deficiency anemia, they're tired and they're fatigued because they don't have iron and they don't have oxygen to bind to that iron. And if they don't have oxygen, how is the mitochondria going to produce energy? So iron is needed for energy, but so is oxygen. So they work together. So red blood cells or erythrocytes are also considered to be biconcave discs, right? You see it's biconcave here, it's concave here, so it's biconcave. The periphery is thicker and the central portion is thinner. So when you have light of a microscope passing through a red blood cell, it usually appears to be lighter in color in the center and darker around the periphery. That gives it a hypochromic color. Hypochromic means less color. So if you look here on the left, all of these are red blood cells. And you can see the center of them is hypochromic. So this is an erythrocyte. Notice on this slide on the left, there's a bunch of cells, but they're not closely packed together like the tiles on a floor. There's lots of extracellular matrix. That's connective tissue. This extracellular matrix in relationship to blood is called plasma, and that makes up 55% of the blood. So that's red blood cells. Now we're going to talk about white blood cells. Now, white blood cells come in two flavors, granular and agranular. Granular refers to the cytoplasm. When you look at this cytoplasm on the right, you can see these grains of salt. That's granular. If we look at the eosinophil, you can see the cytoplasm is granular. If we look at the basophil, we see the cytoplasm again is granular. So those are the three granular sites, and I call them BEN, B-E-N. Basophil, eosinophil, and neutrophil. The term phil, like philos from Greek, means to like. So a basophil likes a basic stain, so it's going to turn this dark. And eosinophil likes an acidic stain. It's going to stain red. And a neutrophil is neutral. Doesn't really take on acidic stain or a basic stain, it's a neutral. So we have a basophil, eosinophil, and neutrophil. These are the three granular sites because the cytoplasm looks very granular. These granulations contain very strong and potent digestive enzymes. So when there's a pathogen or a microbe or some foreign invader that's invaded the cell, this neutrophil can go find it and degranulate, meaning these granules break open and release their strong digestive enzymes to destroy whatever that foreign microbe may be. When you get blood taken and you have a high neutrophil count, they say you have a bacterial infection. If neutrophil counts are high, you have a bacterial infection and your body is producing lots of neutrophils to try and destroy it. Okay, that's what a high neutrophil count is. When you have a high eosinophil count and basophil count, you've got allergies. Your basophil produces lots of histamine, And you know what happens when you have a histamine reaction? Your nose gets stuffy, your scalp gets itchy, your skin gets itchy. That's an allergic response because basophils produce histamine. And the histamine that the basophil produces, the eosinophil tries to neutralize that because the eosinophil makes histamine ACE. Anything that ends in ACE, like histamine ACE is going to break down histamine. That's what enzymes do, they break things down. The other difference to the neutrophils, if you look at the neutrophil and we compare it to the eosinophil, look at the difference between these granules, right? The eosinophil has larger granules, but also compare the nucleus. This has 2 lobes on the right. This has 1, 2, 3, maybe 4 to 5 lobes. So a neutrophil has 3 to 5 lobes to the nucleus. An eosinophil only has 2 lobes of the nucleus. And a basophil has like a kidney bean shaped, but it's sometimes the nucleus is obscured, like it may be hard to see because everything's darkly stained. So again, the 3 granular sites are basophil, eosinophil, and neutrophil. Their differences are the nucleus, where the basophil is kidney bean shaped, it's large, it's obscured, everything's dark staining. The eosinophil is typically bilobed, 2 lobes to the nucleus with large red staining granules. And the neutrophil, although it looks like it's red, they're much smaller, and you have 3 to 5 heads to the neutrophil. The agranular sites are these 2. If you look at the models on the right, whether it's a monocyte or lymphocyte, you don't see granulations, no granules. So we say the lymphocyte and monocyte are agranular. So the white blood cells or leukocytes are granular sites, which is your basophil, eosinophil, and neutrophil, or your agranulocytes, which are your lymphocyte and monocyte. Monocyte is pretty large, no granules, huge, kidney bean shaped. What's unique about monocytes is that they are found in your blood vessels, but they have the potential of diving deep into tissues, but then their names change. They change from a monocyte and they become a macrophage. Macrophage is a very important type of cell. Phage from the word phagocytosis, it means cell eating. So a monocyte can become a macrophage, and a macrophage is like an old school Pac-Man. It just engulfs down debris. It breaks it down. Macrophages have different names depending on the type of tissues you find them in. So if you have a macrophage, if you have a monocyte that became a macrophage in the liver, it's called a Kupffer cell, a Kupffer cell. If it's a monocyte that became a macrophage in the brain, it's called a microglial cell. If it's a monocyte that became a macrophage in the blood vessels to break down damaged cholesterol, they call that a foam cell. If it's a monocyte that became a macrophage in the lungs, it's called an alveolar dust cell. So monocytes become macrophages. They change to a variety of names depending on what organ or tissues you find them in. And you don't have to know all of those names for the exam. I'm just kind of laying down some groundwork for what you're going to be hearing later on in this term. A lymphocyte looks different than a monocyte. Just look at the nucleus, right? The lymphocyte nucleus is as large as the cell, pretty much. And again, it's agranular. When you have a high lymphocyte count, they say you got a virus. So when lymphocyte counts are high, there's a viral infection. When neutrophil count is high, you have a bacterial infection. So the way you can remember that is a little phrase: never bet Las Vegas. Never bet Las Vegas. NBLV. Never bet Las Vegas. NBLV. The N is for neutrophil. The B is for bacterial infection. If the neutrophil count is high, you got a bacterial infection. Las Vegas, LV. Lymphocyte count is high, you have a viral infection. NBLV. Never bet Las Vegas. And then here's the platelet, and the platelet is involved with clotting. So if you want to deactivate platelets, you can take aspirin. You could take warfarin. You could take Coumadin. These are all blood thinners by shutting down the function of platelets. Platelets are designed to help you clot to stop bleeding. When platelets are not functional, you get bruising and you get internal bleeding very, very easy. In fact, Coumadin or warfarin, which people use to thin the blood to try and prevent heart attacks and strokes if people have high cholesterol, this drug's been used for years. It started out, warfarin was used and still is as rat poison. They would kill rats by putting out warfarin, and these rats would die from internal bleeding. So they figured, hey, if we can use this in humans and we just modify the dosage proportionately to their weight, we don't kill people, we just can thin the blood in a safe amount, to a safe degree. Okay, here's a plaque of all the different blood cells. There's a spelling error here to number 4. It should be neutrophils, not N-E-U-R. Take out that R. Okay, so we have red blood cells, which is an erythrocyte. We have lymphocytes and monocytes, 2 and 3. Those are agranular. Then you have neutrophil, eosinophil, and basophil. These are granular sites. Then you have platelets for clotting, and then plasma, which is the extracellular matrix. So when you look at this plaque at first, you see a bunch of cells. And these cells are far apart from each other. They're not closely packed. That's what makes blood connective tissue. So the red blood cells, those should be the easiest to find. That would be number 1. When you look at number 2, these are white blood cells, and the nucleus is just as large as the cells themselves. We said if number 2s, if these are high, there's a viral issue. Those are lymphocytes. 3, these are large cells. Look at those. Those are the kidney bean shells. These are monocytes. They are agranular. We said they become macrophages, but here they are monocytes. Number 4, where are you, number 4? Look at that. You got 1, 2, 3, 4. You have 1, 2, 3, 4 heads to the nucleus. Here, 1, 2, 3, 4, 5 heads to the nucleus. So when there's 3 to 5 heads to the nucleus, that's your neutrophil. That's your first primary responder to bacterial issues. Number 5, where's 5? Here it is, around 3 o'clock. That's your eosinophil. It's 2 lobes to the nucleus. It's granular. That's your eosinophil. And number 6, here's your dark staining granules, the blue ones for basic. That's your basophil, a basic dye, number 6, basophil. The nucleus is somewhat obscured by the dark staining granules. And then the matrix, all this green, the lime green yellowish stuff, that's plasma. That's the extracellular matrix. So now what I want to do is I want to take a look at these and see if we could figure out what type of cell they are. So I'll give you a hint. We'll say number 1 and number 2, these are agranular, and number 3, 4, and 5, those are granular sites. So first off, these are red blood cells, right? Each of these, there are red blood cells with the hypochromic center and the darker periphery. These are biconcave discs. So this large white blood cell that's agranular, huge kidney bean structure, what is that? That would be your monocyte. This one, number 2, agranular, large nucleus about the size of the cell. If this one's high, you're going to have a viral infection, lymphocyte. Now we have 3, 4, and 5. These are granular sites. Let's count the heads to the nucleus. 1, 2, 3, 4, 5. When it's got 3 heads, that's your neutrophil. When these are high, you have a bacterial infection. Let's go to number 5. Hard to see the nucleus. It's very dark stained. It is blue, very dark. This is your basophil. Now we go here. This one's clearly red. It's the only one that's red. These 2 are not. That's your eosinophil, and it's really 2 lobes. Probably not the best to show 2 lobes because it looks like it's 3, but this red cytoplasm is key for an eosinophil. If we look here, here's a neutrophil, 1, 2, 3, 4 lobes. This is a lymphocyte, large nucleus, as large as the cell. Here's a monocyte, that's your large kidney bean structure. Basophil because it's really hard to see. It's just granular, but you can't see the nucleus at all. Here's your eosinophil. It's red, large stained granules, two lobes. That's your This is your eosinophil. These are all red blood cells going around. And here's your neutrophil. One, two, three, four heads to the nucleus. Okay. Okay, that's the end of the chapter there. Let's stop the screen share and stop the recording.